As I promised you in some of the opening comments, we would have some different domains. So here it comes. So our next speaker, Dr. Susanne Bauerschmidt from PharmaLex, is the head of Scientific Data Insights, and she is going to speak to us for a few minutes about the application of ASMs to analytical method validation and use in various regulatory scenarios. So.
Thanks very much for inviting me to give a presentation today here. I'm Susanne Bauerschmidt from PharmaLex. Many of you may know my company from former times. So I've been working at Ostus since several years. Ostus had been acquired by PharmaLex in 2021. And since the beginning of the year, we have been fully integrated. So we are now part of the PharmaLex organization. And I think most of you know that Ostus was the implementation partner of the ADF format. So that's why we are here today. In the beginning, before I start the presentation, I'd like to give a little bit of introduction to PharmaLex, because I think most of you don't know the company yet. So I need to take my laptop. That won't work better. So usual disclaimer. So PharmaLex is a consulting company. We are working in the full space of drug development, and we work on helping companies to develop their products and bring it to the market, and also to help supporting post-marketing activities. PharmaLex itself has also recently been acquired by AmerisourceBergen, which then moved to become a new company, which is now called Syncora. And that's why you find these different kind of names on our presentation slides right now. So we have Syncora PharmaLex, just as a background to the company. So what we are doing mainly is, as I already said, is we work with companies in the discovery preclinical space, go to approval, authorization, growth, and maturity. So this is the area where we do a lot of consultancy. And with the acquisition of Ostus, PharmaLex also acquired two other companies, which are mainly in the statistical space. We together now form the data science and data strategy and quantitative science department within PharmaLex. And what I'm trying to do here today in the presentation is to show how we intend to bring our two different types of expertises together to make use of allotrope standardization in our future projects. So that's the background for the discussion today. And the example I'm going to use is on assay validation. So I'm now going to talk about digitalization or these integration projects, which we've been discussing today in a lot, but the presentation will be more on how do we envision how we could use ASM or other standardization formats in our statistical work. If you think about the business challenge in a manufacturing company, this is an example of using paracetamol, manufacturing paracetamol. So if you do that, you normally have to show that your tablets have what they're intended for. So what you need to validate that the batches have the right amount of compound inside it. For that, you need, in a regulated environment, you need to validate the assay. So you have to ensure that it can detect and quantify the compound, and it can also do that with accuracy and precision. There are some validation aspects on it. So you need to talk, there are some regulations around it, like validation of analytical procedures, FDA procedures, and also USP has some chapters in their compendium to discuss this. So what our colleagues now have been doing is they have created some software that allows to bring the assay validation report that's been generated into the common technical document whenever you do submission to the to the authorities. And you normally do that according to the normal standard validation procedure. So you develop your assay, you develop the experiment you want to do, you do an experiment design, you do the protocol, and then you record data and you bring it back into your protocol. So what we see is that there are several different aspects which you need to consider when you do the assay validation. So it's the range, the quantification limits, the detection limit, accuracy, and precision. And what you do when you define it, so you create the analytical target profile. So that means in our example on paracetamol is that in this case, the procedure would have to be quantify paracetamol in a range from 300 to 500 microgram, for instance. And the distribution of the total error has to be within a certain around maximum uncertainty range, in this case of 2% with 95% of coverage. So you typically do your data collection, you do an experimental design, and that's where now the expertise of our statisticians came in. They have developed a method for experimental design that allows to measure precision, uncertainty, and accuracy at the same time, and to create one experimental design that allows to calculate the uncertainty using the total analytical error. And what they've developed is then a method to create an experimental design that allows to take care of all of this. So here, this would be an example for such a design. Then what they are doing is they get the data and they calculate according to the model which they have defined in the experimental design, the total analytical error, and they automatically produce the reports, which then can be transferred to the regulatory people, who then will take the reports and bring them into the documentation for submission to authorities. So at the left-hand side, you see the method or the way how the method is being used. So you see the range in which the assay is valid, and you can use that as a go/no decision on your performance criteria. So, and what they've been doing is they have developed a small software tool that is in a validated environment, and that supports both the experimental design and the joint statistical analysis of all the results and the automated report generation. And when we had the discussion, so yeah, what could we do with Allotrope, or how can we bring that in? Our idea was that what we could do is, currently the process is really a very manual process, as you have been seeing all over the place in the meeting today. So you have your design of experiments, you do the data in the lab, you do your manual data transfer, in this case, from the lab data into the data analysis tool, you create your validated report, and then you deliver them to the customer. And our idea was now, because what we see is that we have here data break in order to be able to remove the manual data transfer from the lab instruments into that software, that an idea would be that you could use ASM as an integration layer and use tools that you have been developed or which have been shown here throughout the day. Like the previous speaker, they showed their nice model. You showed your model yesterday on the Merck data lake. So how you can bring in that data into a system like that. So I know it is just a very small example, but it's an example how you could bridge from the normal lab into the regulatory space. And that's, yeah, what I wanted to show today. So that would be our idea. You have the same system, you bring in your data using ASM as a data exchange format. You would have a little bit more automation. And I think the other part, which has also been discussed yesterday, you have this dry wet cycle. And the idea could also be that when you have done the design of experiments, you could also use that then to bring in or to try to create the experimental design. So if you have that for your systems in the future, but that I think is something which is a bit more complicated. So that was what I wanted to show today. Yeah, so what the additional advantage of that would be? You wouldn't have manual data extraction anymore. You would like to use that system. The data would be made fair. It would have no break in quality assurance, and it would be state of the art from data quality and standardization. So yeah, short presentation, some ideas on how we think we can bring together our regulatory stats people with our data and data integration people. As one example, I think I just wanted to use that as an idea. Thank you very much.